Okay, everyone, thanks for coming to this afternoon session. While you're finding a seat, I want to start off by playing a little video or a message from one of our sponsors, RTE.
With 100,000 kilometers of power lines and 2,700 substations, RTE, the transmission system operator, provides electricity throughout France. RTE steers electricity, ensuring a real-time balance between supply and demand every second, 24 hours a day, 7 days a week. In order to guarantee a smooth and secure flow of electricity on the grid, RTE operators ensure supply-demand balance from a control room, specifically dispatching. In case of overload, safeguards in the electrical substations alert the operator and ensure the protection of people and property. With the energy transition comes the arrival of intermittent, decentralized suppliers, consumers who can control their own electrical equipment, and local authorities who are fully involved in the national electricity system. The grid must evolve to cope with these unprecedented conditions. To tackle this complexity, RTE is developing a new way of operating the system. The two existing layers, the upper layer of dispatching and the lower layer of substations, RTE adds the middleware, a new layer of intelligence to its electrical system control architecture. The middleware is a platform that centralizes and processes data through embedded applications. It is a veritable brain covering a geographical area of about 10 substations. For example, the zonal automaton application. The middleware collects data only from the concerned zone and distributes the relevant information. The automaton can then autonomously activate, if necessary, the most relevant flexibility levers in the area, including switchgear in substations, wind generation, or battery charging. A regular dialogue with the dispatching center allows the middleware to configure itself and coordinate the other regions. This 4.0 architecture, both decentralized and hierarchical, will constitute a cyber-physical system of systems. In short, a hybrid system, combining cyber or digital intelligence and the physical system through the grid's infrastructures. This new architecture offers a generic solution that is efficient, resilient, robust, and agile, enabling the optimization of renewable energy integration both today and in the long term. In this way, RTE secures a head start to better support the energy transition.
Now, we're ready to introduce our next keynote speaker, Mary Dunlop from Boston University. So Mary is a professor of biomedical engineering at BU. She has a BSc in mechanical and aerospace engineering from Princeton and a PhD from Caltech, where she studies synthetic biology with a focus on dynamics and feedback and gene regulation. Now, her lab engineers novel synthetic feedback control systems and studies naturally occurring examples of feedback in gene regulation. And what she's here to talk to us about today is one of the most exciting applications of machine learning and control into biology. She'll talk about a new approach for controlling gene expression in single cells that integrates ideas from machine learning control theory in biology with profound applications in biomedicine and beyond. Mary.
All right, thank you. Well, I am really pleased to be here today. I'm going to tell you about some of the work that my group has been doing to use light to control, coupled with feedback to control gene expression in individual bacteria. And I recognize that this is a topic that is fairly different from what you've been hearing about so far. I think you will find it exciting and interesting. And what I need to do first is give you a little bit of background on why we're interested in this problem and tell you about some of the biological challenges and applications that we're working on, and then we'll get into some of the feedback control things that we've worked on. Okay, so I'm going to start out by showing you two movies here, and what you're looking at are E. coli bacteria. They are trapped in a microfluidic device, so that rectangle in the middle is about a micron deep, and the cells are constrained within it to grow in a single layer. in 2 dimensions. And then at the top and bottom of this image, there are deeper channels through which we can flow fresh growth medium to provide nutrients to the cells. But in these experiments, we also introduce a short pulse of a lethal dose of carbenicillin. So carbenicillin is a beta-lactam antibiotic. It's similar to things that you may have heard of, like ampicillin or penicillin, and it works by interrupting cell wall biosynthesis. And when we conduct these experiments, the cells are growing well, we introduce the carbenicillin, and then you can see these like lysed remnants of cells because their cell wall has been disrupted, and by the end of this movie, nothing is growing in this trap, which is, of course, what you would want to have happen if you were treating a bacterial infection. You'd like to have everything killed off. But we run these experiments in parallel in these microfluidic chips where we have many different chambers arrayed in parallel, and so we can take these cells, which are all genetically identical, and look for things that can happen that are different. And every now and then we see something like this movie on the right, where the cells are growing well. We add the antibiotics, most things are killed off, and then there's this one cell in the middle, and after the antibiotics have been removed, it's able to grow and divide, and it completely recolonizes the trap within the course of just a few hours. And so we've been really fascinated by looking at these cell-to-cell differences in antibiotic resistance. And when you think about antibiotic resistance, you're probably thinking about sort of genetic changes due to either a mutation or perhaps horizontal gene transfer. And that's the classic way that bacteria can evade drug treatment, but it turns out that research in the past sort of 10 or 20 years has identified a number of different mechanisms by which cells can temporarily evade drug treatment. And these are really subtle in that an individual bacterium will enter a drug-sensitive state and then a drug-resistant state and then go back to being drug-sensitive. And it is consequently very hard to measure these things happening because you can't just do common things like sequencing to identify what state these cells are in. And so they temporarily transition into these states that we'd like to avoid. Of course, if you're treating an infection, you don't want this condition on the right to happen. You'd like cells to all be killed by the antibiotic treatment. And so it's well known in the field that gene expression is stochastic. There's variation that occurs due to bursts in transcription and translation and a number of other factors as well. In order to see this, we need to be able to look at properties of the cell. And using microscopy, you can, of course, image things like the growth rate. But a common trick in the field is to use fluorescent reporters. So these come from other organisms that can light up. So I like to think about examples of luminescence, like, for example, fireflies or lightning bugs can light up. There are things that come from marine organisms like jellyfish that encode green fluorescent protein, as we're using in this case. And here we're using the gene encoding a fluorescent protein to report on expression of a particular transcription factor, which is called GADX. So GADX controls dozens of different downstream genes within bacteria that regulate stress response. And if we look at GADX expression in individual cells, then we see this histogram distribution of expression levels, and you can see it in the images on the right. So the phase contrast image is basically just a picture of the cells, and then the fluorescence image shows you the GADX expression, and hopefully you can appreciate that some of those cells are brighter and others are dimmer in terms of their expression of this particular regulatory protein that controls a stress response and in bacteria. And so where does this come from? There are a variety of different ways that this variation can arise. One that's relatively straightforward to understand is that these are very small microorganisms. They're about a micron on the shorter dimension. So you're talking about 10 to the minus 15th liters, a femtoliter in volume. And consequently, you can have very small numbers of molecules that play important roles. So for example, you might have 10 molecules that play a particular role in stress response. And when the cell divides as it's growing, sometimes that's 5 and 5, but sometimes it's 3 and 7, and sometimes it's 2 and 8. And this partitioning in cell division can introduce variability. There's also many other sources of noise that arise. A common one that bacteria use is that they encode feedback networks that purposely drive the generation of variability. For example, making multi-stable networks such that you have multiple cell states in order to hedge against uncertainty in the environment. There are also delays that exist between sensing an environmental change and the time it takes to make a protein, the process of transcription and translation. and that can vary across cells. There are also global factors like changes in nutrient supply, temperature, or other extrinsic factors that are at play here. And so I'm going to show you some data where we're measuring these single-cell gene expression dynamics. But before I do that, I need to tell you a little bit of methodological details. So we work, in the rest of this talk, we're going to be working with this microfluidic device that we use a lot in our lab, where bacteria are constrained to grow in these single-file lines. This particular device is often referred to as the mother machine. The cell at the top is the mother cell, and then as it grows and divides, those are daughter cells that enter the chamber and are pushed out and then swept out with the fresh media. I'm showing 3 different chambers here, but in practice there are thousands of these arrayed in parallel lines, which makes it really nice for imaging. And the trick with this is that the mother cell stays at the top of the chamber, and so you get to image that one cell over many hours, whereas ordinarily it would grow and divide and then be kind of lost in a larger ship. Okay, so here I'm going to show you a movie of GADX dynamics over time. So the fluorescence images are on the right. And what you're watching here is, you'll see that GADX is, it starts out being kind of uniform across this lineage, it dips down a little bit, and then over time you'll see that GADX starts to increase, and you go through this kind of pulse of GADX expression, and then it decreases again. You're seeing the mother cell at the top growing and dividing, and then it's sort of low again, and then GADX will again increase. And we get a lot of data like this. I don't have time to talk about the details, but it's actually an interesting feedback network in the gene regulatory pathway that produces these pulses in GADX expression. And if we look at fluorescence over time, so GFP is green fluorescent protein, and then for one cell like this, then what we see is that it goes up, it goes down, and it goes up, and it goes down again. We can summarize this with a heat map with the blue and yellow traces, and then I've shown 25 representative cells here over the course of 15 hours. And hopefully what's apparent is that there's a lot of variation in GADX levels across individual bacteria. Some of them are producing a pulse here and others producing a pulse there. It's not synchronized in any way across the bacteria that we're looking at here. And so our next question was, GADX is this regulatory protein that's involved in stress response, but does it actually have phenotypic consequences? Like, is there any reason to have this happen? And we tested this by doing a similar experiment to what I showed you at the beginning, where we then introduced a pulse of a lethal dose of ciprofloxacin. Ciprofloxacin is a fluoroquinolone antibiotic, so it's a slightly different class than what I talked about previously. But in this experiment, you're looking at 3 chambers within the mother machine. And the cells are all growing well. They have differences in GADX expression. At hour 4, the antibiotic will go in. And after that, you start to see differences. They're most apparent in those phase contrast images where the 2 chambers on the left start to sort of disintegrate and those cells have been killed by antibiotic treatment. But the one on the right does something very different. That cell grows and divides and then it returns to its normal GADX pulsing dynamics. And so we've been really fascinated in my group by asking, why did that one chamber on the right have cells survive where the other 2 died? These are genetically identical cells. Why is it that this one bacterium was able to evade antibiotic treatment? Was it something about GADX expression in particular? You may have noticed that those cells are brighter green. Or is it something related to the growth rate? Antibiotics are well known to be sensitive to how quickly bacteria are growing, since many of the processes target cell growth. Or was it just something about random chance? And so we can do experiments like this where we then retroactively look at which cells survived and which ones died, and then look back in time and look at GADX expression levels that preceded survival or death. And what we find when we do this is that the cells that are in the midst of a pulse of GADX expression are much more likely to survive than those that died. Those that pulsed in the past but weren't pulsing at the time of antibiotic addition are less likely to survive. And those that never pulsed at all very commonly die. And so we and others in the field have been kind of largely in the state for a while, where we are observing cells and asking questions about, correlation-based things. Is this likely to correlate with survival or not? The project that I'll tell you about today kind of flips that story around and asks, what if we could precisely control gene expression in individual cells? Rather than sort of sitting back and waiting for it to happen, can we actually drive cells to follow particular, GAD-X expression patterns, and then ask questions about whether this is linked to antibiotic survival, so that we could much more directly test things? This could help us reproduce natural expression dynamics, so we could ask, for example, how long does that pulse have to be to enable survival? What kind of amplitude is required? And we could also take advantage of the fact that some of these events are very rare, and if we can make them happen ourselves by forcing the cells to follow specific patterns, then we can get a much more accurate picture of what's going on in these very rare and uncommon circumstances. Okay, and so the project that I'll tell you about is on developing a method for doing optogenetic control of gene expression in single cells. So optogenetics is using light to control gene expression. And this project was really pioneered by a phenomenal postdoc in my group, Jean-Baptiste Lugagna. And in it, he took advantage of this particular two-component system called CCA-SR, which is a light-responsive system. So this was developed by Jeff Tabor's lab at Rice University a number of years ago. And it's derived from cyanobacteria, which are photosynthetic organisms, and so they have genetic parts in them that are responsible for sensing light. We can take those genetic elements and then put the DNA from cyanobacteria into E. coli, and then make them light responsive. And this is an oversimplification, but I think a useful one. I always think about this like a traffic light or a stoplight. Green is on and red is off. And so in this original paper, what the Tabor lab did was they connected it to expression of green fluorescent protein. And you can see in the image on the right, when you shine green light on the cells, they turn on their fluorescence. Red light turns it off, green light turns it on, and red light turns it back off again. So the first element that we're getting here is really exquisite temporal control. For those of you who have done anything with working with biological systems, which may not be a huge number of people in this audience, this is important because I think the typical way of doing this is by adding chemical inducers, and those are kind of slow to diffuse. You have to wash them out. It's much harder to do this sort of fast temporal control, and light offers great advantages here. The other fun thing about working with light is that you can spatially pattern it. And we do this using something called the digital micromirror device, which is the same kind of technology that gets used in modern projector systems. We couple that with our microscope, and that allows us to pattern spatially on the field of view of the microscope that we're working with, so that we can individually target chambers within the mother machine with either red light or green light. And so what we did in this project was that we designed a feedback controller that allowed us to try to use red and green light to kind of push cells where we wanted them to go. And here's the kind of simple block diagram version of this. So ideally, we'd like to be able to have some user-defined objective function. I've drawn a sine wave, but if we're talking about GAD-X expression, maybe we're interested in some sort of pulsatile dynamics. So it's something that's user-defined, and then of course, if this works well, what we'd like to have our cells do is follow that objective with their fluorescence values. So you'd like to see the cells get brighter, and then dimmer, and then brighter, and then dimmer. And I've already talked about the like cell element of this block diagram here. We're shining red and green light on the cells, and then they're changing their fluorescence in response. We'll use some image analysis to process the microscopy images on the fly, compare the actual fluorescence that the cell is exhibiting to where we'd like it to be, and then make some sort of control decision to decide whether to apply red or green light. And so the first thing that I'll focus on is this question, how do we adjust those red or green lights to compensate for the difference between the objective and the actual GFP levels? And I'll show you the version 0 of our design. So this is the first thing that we built. And here we didn't even try sine waves. We just said, let's just do a step input. We'd like GFP, green fluorescent protein, to start low and then go to some higher set point value. And of course, you're well aware that the step response shouldn't be instantaneous. Like, just to set your expectations, transcription, translation probably is going to take half an hour to an hour to get us up to GFP levels that are where we'd like to see them. But ideally, we'll see a nice step response from this. The movie on the bottom shows 3 different chambers within the mother machine device. The cells that we're trying to control are those mother cells, which are at the top of the image, and they're outlined in different colors, either blue, cyan, or yellow. And those correspond to the plots on the dots on the plot on the right-hand side that show what the fluorescence levels are over time. And then the last thing you need to know is that the color of light we're shining on the individual chambers is shown by the rectangle at the top of the mother machine image. So right now, all those cells are above where they should be, so we'll shine red light on them. The fluorescence levels drop in those individual bacteria, and everything is looking good. About hour 4, things will turn back on. We shine green light on the cells, and then the controller adjusts dynamically between red and green light based on whether the cell is above where it needs to be or below where it needs to be. And we're tracking this set point here and fluctuating kind of back and forth between red and green light. And so when we got this working in the lab, we were absolutely thrilled because it means we got, image analysis working on the fly, no small feat. It means that we, are doing single cell resolution control in a microfluidic device with individual bacteria, measuring fluorescence in real time, and we were like super excited about this. But I think for this audience, you're probably underwhelmed with our step responses, and I will concede that point. So basically, we were just doing like bang bang control there. We were just doing either red light or green light. That's it. And there are these really there are long delays here. This is a terrible control design, and so there are other things that we could do that are more nuanced. In fact, people in the field have done the equivalent of proportional integral control, where they've taken not just red or green light, but done some sort of either combination of the two, or maybe use a dimmer version of red light. There are lots of ways to implement this. The current state of the art was to do model predictive control, and there have been a small number of labs that have employed this to control. To date, it was or before the work that we did, it was a few dozen cells was the limit of what they could achieve with those controllers. And so the MPC style approaches do work well for this. In fact, they work a lot better than the proportional integral implementations that were out there before. But they're, again, limited in the capacity to work with more than just sort of a very small number of cells. And so when people have implemented this kind of thing in the past, just to put it in the context of this biological system, what we're asking here is, if we have a model, and traditionally this is an ordinary differential equation model, I've put up an example there of the type of system of equations that we might use to do this. So usually, maybe a couple differential equations that describe some elements of that 2-component system, the optogenetic system, and then also the fluorescence change in response. And then what these models do is you would ask it, okay, if I were to apply different candidate light stimulation strategies, like either green, 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 red light would be the top one, or red, green is the middle one, or red, 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 then you would run that model and then look at what the GFP level is predicted to be, and then you would select the light stimulation strategy, in this case the middle one that most closely matches that objective function. And when we do this, it's typically performed as a receding horizon control. So the model will predict a future over a typical window for us is about a 2-hour horizon. We've tested other variations on this as well. And control is reevaluated and updated regularly. We do this every 5 minutes. We run into technical limitations with cells where we can't image them all that frequently. About 5 minutes is as fast as we can go, especially in fluorescence, because you start to run into cytotoxicity issues with that kind of resolution if it increases too much more. So the current control implementation works well for a small number of cells, but it's relatively computationally expensive, as I'll show you later on. And it also requires an expert-defined model. So to write down these differential equations, yes, they're not that complicated, but you do have to know something about transcription and translation and the relative rates of reactions, and that limits this to systems that you actually know a reasonable amount about, which ends up being a pretty big constraint for biological systems where we often don't know much about the properties involved. It also is a one-size-fits-all model, so some cells are very touchy and extremely sensitive to changes in light, and others are very, very sluggish and not very responsive. And we could increase the complexity of this model to handle that, but that, again, comes at a computational cost. And so the approach that we did was we said we just replaced the differential equation model with a deep learning model in a deep model predictive control style approach. I'll show you that this is both faster and more accurate than the traditional model predictive control that had been the current state of the art. It's also agnostic to the system, so we aren't telling it anything about these being, E. coli bacteria. We're not telling it anything about, the particular reaction rates or anything about what's going on. We just feed the data to the model and it learns. And then we can incorporate many features as inputs. So the classical way of doing the model predictive control would use the fluorescence, but here we can incorporate other things that such as cell length might tell us something about growth rate, but also things that would not traditionally be included, like the image sharpness is a really good example. So is that particular frame of the microscopy data in focus or not? That's relevant for telling us how high quality that particular data point is. But of course, as with all of these data-driven methods that use deep neural networks, you need a large training set to work with these. And this is where it's a real advantage that we're working with these microfluidic devices where we have many cells arrayed in parallel, and we can customize our light inputs on them. And so what we do in these experiments to gather training data is that we acquire these data set of single cell responses where we shine randomized patterns of red and green light on the chambers within the mother machine. So here's what it looks like. We start out with a 3-hour equilibration phase with just red light. And then after that, we switch to randomized fluctuations back and forth between red and green light. And then we record the fluorescence coming off of the bacteria, and then also things like cell length, the focus score, the microscope, et cetera. And so we acquired the training data, and it's just shy of 16,000 different single cell traces in response to these light inputs. We can do this we can do about 4,000 cells at once in these experiments. So this was 4 different experiments, each 24 hours in length. So in under a week, we were able to acquire the training data for this, for these responses. And I think it's kind of, maybe just pause for a moment. We can look at that image on the left-hand side together. So what you can see here is the fluorescence in response to these red and green lights. And one of the things that you'll see is that the system is much more responsive to green. Like green light will turn it on and it turns on really rapidly. When red light goes on, it turns off much more slowly. And that's because these cells are not actively degrading the proteins involved. And so the only way they go away is when the cell grows and divides, then it partitions the proteins that are relevant, and then you end up with a sort of effective dilution of those proteins over time, but it's much slower than the on rates of the system. Okay, so we take the data that we got from these experiments, and then I'll tell you a little bit about the model structure and inputs that we use. So what we do is we artificially partition the data into past and future, and we feed it the model the past data, and then ask it to predict what will happen in the future if we apply red or green lights. And so we take that past data, actually let me just go back for a moment. There are 8 different features that we look at here. Each of these are time series data, and the 8 different features include the red and green light optogenetic stimulations, the fluorescence of the individual bacteria, the area of the cell, and then we threw in a bunch of other things that we kind of weren't sure if they would matter or not. For example, like the mean chamber fluorescence of all the cells in that chamber, the standard deviation of it, the number of cells in the chamber, does that matter? I don't know. And then things like the neighboring stimulations. There may be light pollution effects, and we have all of these arrayed in single file lines, like the neighboring light that the chambers are getting might matter. And then we have image sharpness, which is a surrogate for the focus score of the microscope. Okay, so we take the data and partition it into past and future. We take the past data and feed it into a long short-term memory encoder, which generates this small latent space that tells us about the past of the cell. We then take the candidate light stimulation strategy for the future and then feed those 2 parts together into a multi-layer perceptron. That's used to predict the time series in the future over the horizon we're interested in. And then we compare that to the ground truth, the actual measured data, calculate the root mean squared error between those 2, and then try to minimize that when we're doing the training. Okay, so here's what the performance looks like on test data that the model's never seen before. This is a histogram on the left-hand side of the root mean squared error. Of course, error we'd like to minimize, so lower is better. To give you a sense of what this actually looks like, I've called out 3 different values, the 25th percentile, the median, and the 75th percentile. And you can see examples of these from top to bottom. So the 25th percentile, what you'll see is that the model prediction and the actual GFP signals are nearly identical. Even the median prediction is excellent as well. And then for the 75th percentile, you can see that it's still quite a high quality prediction, but it's not surprisingly, it's better in the near term than it is for forecasting out further into the future. But overall, we were really pleased with how well this model did at predicting these stochastic gene expression dynamics just from data alone. And I'll talk a little bit about accuracy and computation time. So we compared our deep model predictive control approach to the standard ordinary differential equation style MPC approach. And what we found was that the error, which is the plot on the left, was slightly lower for the deep MPC approach. And this probably comes from the fact that we're feeding it a bunch of information that the standard differential equation model doesn't have. But it's not really a dramatic difference between the two of them. The really sizable difference comes in computation time. So this is, the plot on the right is a log scale on the y-axis, and what you can see is that the deep model predictive control strategy is 4 orders of magnitude faster than the equivalent ordinary differential equation approach. And in practical terms, what this means is that we can run many, many more cells with this, control many, many more cells, and test, dramatically increase the throughput of our approach here because we have a much faster computation time. Okay. So I'm going to just tell you one slide worth of information about what we do for image analysis. This was a really critical part, and it was actually where we first started with this project, because when we started building this feedback control loop, we were able to get cells to grow in the mother machine, but there was no way to automatically detect where bacteria were within the mother machine and do this on the fly. And so we needed to be able to obtain these high-quality single-cell level data from time-lapse microscopy images. And the computer vision field has come, a really long way, especially in cell tracking. But at the time, this just didn't exist in any kind of form that was usable for us. And so we developed this algorithm that we call Delta, which stands for Deep Learning for Time-Lapse Analysis. It uses a series of convolutional neural networks to segment, track, and then reconstruct lineages from time-lapse images. So here's an example of what it looks like for cells growing in the mother machine. I don't know if you can see that there are little circle outlines highlighting where the cells are. They're white arrows that appear every time a cell divides. And this algorithm does a beautiful job of tracking individual bacteria over time. And we, of course, need this to be very accurate and rapid, because if we're running this in the loop, in the feedback control loop, we can't have a lot of errors in that, and we certainly can't have a human sitting there and drawing little circles around cells, which is probably what you would have done maybe 6 years ago. We have also tested this out on other types of geometries and other cell types. This is Bacillus subtilis growing in a microfluidic chip. And this is a kind of setup where there are thousands of bacteria that you need to track. And we were able to run our Delta algorithm on this without any retraining. And it does a beautiful job. It's not perfect. There's like a circular post in the middle of that microfluidic device that it calls a cell, but it wasn't retrained on this. And we were really pleased kind of with the generalizability of the Delta software. And I've mentioned a couple of I've shown a couple of papers here if you're interested in learning more about this, but the code is all available open source as well, of course. Okay, so to bring this back to the feedback loop, connecting everything together in this architecture, what we would like to do is to set some objective function. So here I'll return to sine waves. And we'd like to be able to push cells kind of back and forth with red and green light to follow this objective function. And what the model predictive control is going to be doing is it will evaluate candidate light stimulation strategies. There are 3 shown there, but in practice we use 1,000 of them for every cell that we work with. And ideally what it'll do is come up with some sort of optimal light stimulation strategy such that the model predicts that the fluorescence will follow the objective function. We select that, apply it, and then continue on. Okay, and so here's what it looks like. I'm going to play the movie in a moment, but I'll get you oriented first. There are 3 different cells here, each tracking the same sinusoidal objective function. The top is 25th percentile, middle is median, and bottom is 75th percentile. Maybe focus your attention on the middle one for now. And Here, we go through this initial 3-hour period of just red light exposure before we start tracking the objective. Everything before that gray line is the model prediction. What's behind is following that, is what actually happened. And so you can see we're able to push these cells back and forth with the red and green light to follow the fluorescence levels. Maybe drop your attention to the bottom one, the 75th percentile. So here the control continues working, but this is something we see quite frequently. The bacterium is just not responding the way we want it to. We're doing the right thing. We're applying like green light, green light, green light, and it just is decreasing. And then for reasons I do not understand, it just picks back up again and starts behaving. And so we see these examples, pretty frequently within the data. And one thing that's kind of interesting to look at from this is that all 3 of these cells we're trying to track the same objective function, but if you look at the red and green lights in the background that you needed to apply to make this happen, they're very different, highlighting how unique each of these cells are. The one on the top needed a lot of green light in the beginning to get going, whereas the one in the middle just needed like kind of little flashes of green light to make this work. And I talked a lot about the speed with which we can do this. And so we, of course, didn't just control 3 bacteria. What we were interested in doing was actually controlling a lot more than that. In this case, we controlled 10,000 different cells. We did this over the course of 3 different experiments. And 10,000 cells is a lot of data. And so to visually represent this, what I've done is tiled it into a 100 by 100 pixel grid, but each pixel corresponds to a single chamber within the mother machine device. And each pixel gets a customized objective function, and here we're using phase-shifted sine waves so that we get a pattern. So initially, there's a 3-hour period with just red light exposure, and then you'll see the objective function appear on the left. So we get these expanding concentric sine waves caused by the shift in the phase shift, and then the middle one is the actual fluorescence measurements in individual bacteria for 10,000 different cells over the course of, I think it's something like 18 hours total of data. And you'll notice that image has a little bit of like fuzziness to it, and some of that comes from the fact that we're just working with real bacteria. So a black pixel is literally a dead pixel. There's a dead cell there. And then some of the white pixels correspond to cells that have filamented and they're kind of long and unhealthy. We didn't do any data curation for this. It would be, of course, easy to do that, but we were sort of fully interested in fully representing what would happen with this type of control. So you're seeing all of the data there. So we were really pleased with our ability to use these customized objective functions. And of course, we're not limited to sine waves, and Jean-Baptiste had this fun idea that we would pull a clip from, here, I'll just start playing this, from 2001 Space Odyssey, this epic Stanley Kubrick film from the 1960s. So after 3 hours, you'll see this appear. That's the objective function on the left-hand side. On the right-hand side, you see the corresponding fluorescence values, and you can see that we're doing a really beautiful job of tracking this. We can accurately track these complex dynamic objective functions, but rapid transitions are really hard. So I mentioned that going from light to dark, so going from like something that's brighter and applying red light, that's slow. So those kinds of transitions are harder for us with this system. But overall, we were really pleased with our ability to track these complex functions in 10,000 bacteria, which is really unprecedented for the field. So let's talk a little bit about some of these results. So one of the things that we found is that that growth rate really strongly influences our tracking fidelity. So if we plot actually just from the data, you probably saw that the quality kind of degraded over time as we got like further and further into the movie. But if we plot the absolute error as a function of time, you can see that that we do a decent job up to about 24 hours, and then after that, our error starts to creep up. And this comes from the fact that the cells are getting a little bit unhealthy. When we're trapping them in the mother machine, that these cells at the end basically age. There's a sort of aging effect where over time those get to be very, very old and they don't perform nearly as well. We can see this as a decrease in the overall growth rate, which is that pink line, and also an increase in the fraction of cells that are over a certain length, which is basically just showing the ones that are filamenting. It's not that all the cells start to grow a little bit more slowly. Instead, what actually happens is you get this bimodal distribution where the cells are either healthy or they have stopped growing. And as you progress through time, this distribution of healthy cells shifts more towards this distribution of unhealthy, non-growing cells. And one thing that was kind of interesting for us was that we plotted how well, what the error was as a function of the growth rate. And not surprisingly, if the cells are growing very slowly, which is on the left-hand side of that bottom right plot, then your errors are high. What was surprising to us at first was that on the right-hand side of this plot, that when the cells are growing really rapidly, we also got high errors there too. And in retrospect, this made a lot of sense, because if you're in a situation where the cell is growing really rapidly, it's just actually diluting things so quickly you can't keep up with it. And so then our control fidelity is pretty low there as well, but there's a sweet spot in the middle of normal growth rates where we can achieve good control. Okay, and everything I've told you about so far is just controlling a fluorescent protein, which we can do super fun things with. We can reproduce movies, but we were, of course, interested in extending this to other applications as well. And so I'll tell you about some steps in this direction. So a talented PhD student in the group, Caroline Blasick, worked with Jean-Baptiste to extend the system to control not just green fluorescent protein, but expression of TET-A, which is an efflux pump that can export tetracycline. We coupled this with expression of green fluorescent protein so that we could measure the fluorescence levels, but then know what was happening with the tetracycline efflux pump levels. And what we did in this was we didn't try to do anything fancy with dynamic signals yet, though we'd like to go there. We just did some step changes. We have two kind of open loop versions of the control, constant red light and constant green light, which set the bounds of what we can achieve. And then we set 3 intermediate points that we wanted to follow. Objective functions that hit at 800, 1200, or 1800 arbitrary units of fluorescence. And you can see from the plot on the left-hand side that we're able to really nail those values that we'd like to achieve with this model predictive control style of approach. And there's this subtle decrease in growth rate, which you can see from the data on the right. But interestingly, if you look at the growth rate on the right, you can see that the more the cells are expressing this efflux pump, the worse they grow. So there are these very subtle differences in the cost of this pump that would be obscured by most measurements. And setting them very precisely here allows us to carefully distinguish between expression levels and growth rate. And then at 9 hours in, we add a lethal dose of tetracycline. The control quality really degrades at that point because the cells are not growing well anymore, but you see this inversion in the tetracycline survival where the growth rate for the green cells continues on. They're impacted, but they keep growing. And then the ones that had low efflux pump expression really just die off almost immediately. To look at this another way, if you look at the percentage of the population that remains growing, the cells that have higher levels of TET-A expression survive for longer. And because we have all the single cell data, we can look at this happening in real time or over the course of this experiment. And so these are violin plots showing the growth rate as a function of time, and the different TET-A levels from left to right are increasing in their levels. And then after we add, at 9 hours, we're going to add tetracycline, and you'll see that there's a drop in the growth rates, but that it corresponds really perfectly to the different TET-A expression levels that we've set. And then you end up with these distributions that have both growing and dying cells in them for the higher expression of the efflux pump. Okay, and then finally, just as another application, kind of illustrating where we're going with this, Caroline worked on extending this not just to one gene, but to controlling parts of a regulatory cascade. And she worked with this PQ system, which is a 2-component system that is very involved in stress response and survival of antimicrobial peptides. And the difference here is that rather than just controlling GFP or TET-A, we're controlling a regulatory protein that now controls lots and lots of different downstream genes. And so we can look at input and output relationships if we use 2 different fluorescent proteins. We use M-cherry, which is a red fluorescent protein, to measure P, and then we look at the suite of downstream regulated reporters using green fluorescent protein. And so there are many, many different genes we can look at. And in each case, here she's using differences in the amount of green light, and you can see that the input increases commensurate with the amount of green light, and then the output changes. It's different for each of the different downstream genes we looked at, so we can look at signal propagation within these networks now, and also single cell level responses. And we've been able to do similar experiments to the one that I described before, where we have differences in antimicrobial peptide survival as a function of the input and the output, which we can measure simultaneously, which is allowing us to look at how signals get propagated within these networks now. Okay, I want to thank the people who did this work. I highlighted both Caroline's work, and then Jean-Baptiste was really the major inspiration behind this, so I want to make sure that I call him out again. And I will thank you for your attention, and I'm happy to take questions.
Thanks, Mary, for a very exciting talk. So questions? There's a question there. Kula, can you pass the microphone?
Thank you very much for the fascinating talk. I've got many, but I'll just ask you. One is about the model that you used. So basically, you use training set to train your model on these thousands of cells. But have you thought of like adjusting then the model online on the cell you're observing? And then the second question is related to instead of using like an MPC strategy to control the cell, have you tried to simulate with light inputs like a regulatory pathway that could then implement it to the cell, like prototyping kind of, in a prototyping kind of fashion?
Let's see, let me answer your first question, and then I will come back to the second one because I might need to clarify. So the first question was related to the, sorry, remind me, the model was, yeah, say it again.
So do you have one model for all of the cells?
Oh, yes, okay. Right, are we doing anything where we can like update as we're going in the experiment? So we didn't in this case. I think that you could certainly take advantage of things where you're updating on the fly as you learn more about that cell or about the responsiveness generally of cells. We have not done that, but I think it's certainly an exciting area to pursue. And then can you ask the second one again?
Yeah. Sorry. Your control strategy is an MPC strategy. So another way to control cells in literature is like by embedding some regulatory network within the cell. So as a way to prototype and testing this before implementing the pathway, you can use light to basically replicate or mimic the pathway.
Yes, I love this idea. So, okay, I think one of the things that's really fun about working with light is that as engineers, we can work with it really easily. So the other ways of implementing stuff, feedback control laws in bacteria would be, you might go into the lab and, clone some parts where you have to order the DNA from add gene, and then you get it, and a week later it arrives, and then you have to PCR it, and then you amplify, and if you're good, 3 months later you have this built. And so that's a lot of the kind of classic like development of feedback control systems in synthetic biology has used that kind of design, build, test, learn cycle. And I think one of the really exciting things about working with light and putting some of the control outside of the cell is that you can actually do prototyping and interfacing with cells in a much more straightforward fashion because we can use these like signals that are kind of human interpretable and straightforward to program. And so, Mustafa Kamash has done a lot of really nice work in this space as well. But I think that one of the real advantages of doing this kind of like hybrid approaches is that you could rapidly prototype things that you then learn design principles from, and then you would understand, oh, okay, this needs to be very responsive, and so I'm going to pick a particular promoter that will have that kind of attribute. Then you can really refine your search space such that when you're building things in the lab, you can do something very targeted rather than having to test many, many iterations of the design using a very slow process.
Thank you very much.
Okay, one more question perhaps, because we need to move on. We need that if we want to take it.
Thank you for your very interesting talk. The question is, how is sensitive your deep MPC to the changing of, parameters of biological parameters like temperature? I don't know, what can change?
Okay, yeah, so I had thought you were going to go a different direction with that question, but I will, so that the temperature and other, biological components, it's actually not that sensitive, and we know that because we didn't, when we switched from GFP to TET-A with GFP, and then also to the FOPQ system, we didn't have to retrain the model each time, and so we are actually able to get pretty good data that works, a pretty good model that works across a variety of different conditions, and it's not that we have to sort of update things very regularly. It's quite robust, yeah.
Okay, so it's just the matter of your physics, let's say, or biology
Yeah, no, I
nature.
think just really quickly, the caveat to that is that I think if we did something like controlled a bistable network or did something that was very different in terms of its dynamics, then I think we would probably need to do something more complicated. Yeah.
Thank you.
Okay,
Thank you.
let's thank Mary again for an exciting keynote.